Hi, I'm Camilla Martinez, and I'm going to be talking about the genetic factors in the evolution of Ostracod bioluminescence mating displays. This is an ostracod. Ostracods are small marine crustaceans that produce bioluminescence for two reasons. First, to deter predators, and second, to attract a mate. Different species of ostracods have different mating displays. They vary in direction, pulse size, pulse direction, and time. By studying the evolution of ostracod mating displays, we can better understand how behavioral phenotypes evolve and change in different species. Ostracods produce bioluminescent packets of mucus with the enzyme C-luciferase, which produces light when reacting with the substrate vargulin, which is a high-energy substrate. In previous studies, the function of C-luciferase has been negatively and non-linearly correlated to the pulse length of ostracod mating displays. In this experiment, I studied two species of ostracods, P. morini and P. incoinae. P. morini have short bursts at 150 milliseconds per burst, and P. incoinae have longer bursts at 350 milliseconds. Although these have different mating displays, they only differ in 11 amino acids. This is the sequence of C-luciferase. It's 554 amino acids long. However, there are only 11 amino acid divergences. And I chose site 32 and site 351 to study. The cysteines are highlighted in yellow, and we picked these sites because of their proximity to cysteine. Cysteine forms disulfide bonds, which are crucial to protein folding. And the protein folding, when it's affected, it can affect the efficiency of the C-luciferase enzyme. The mutation site can be shown by the red star, and we manipulated P. morini's amino acid sequence to match that of P. incoinae's, one amino acid at a time, to find the effect of changing that amino acid. In site 32, we changed alanine to valine by switching the codon sequence from GCG to GTG. For site 351, we manipulated the amino acid sequence by changing the valine to an isoleucine, changing the codon sequence from a GTA to an ATA. Prior to our study, DNA had already been isolated from P. morini and put into E. coli by heat shock. They were then grown, and we isolated the plasmid from this E. coli. We took this template DNA and the primers that we designed with the mutations embedded in them. We then did mutagenic PCR to amplify this DNA. We then used these new transformed plasmids to transform E. coli by heat shock. We grew them on plates, and we chose a couple of colonies from each plate to screen. This was our first attempt at mutagenic PCR. The first lane shows a 1 kilobase paired DNA ladder, and the second lane is mutation site 351. The next lane shows the negative control for 351. The fourth lane, in which you can see a slight band, is mutation 32, and the negative control for 32. This attempt at mutagenic PCR was not super successful because there were no bands shown in the mutation 351, and there's a slight band at mutation 32. So we decided to run these PCRs at a temperature gradient, and we varied them by concentrations of DNA as well. The first lane, once again, is a 1 kilobase paired DNA ladder, and the next 8 samples are 20 nanograms run at a temperature gradient from 58 degrees to 67 degrees. The first 8 are 20 nanograms of DNA for mutation site 351. The next 8 are 2 nanograms for mutation site 351, and the next 8 are the negative controls for mutation site 351. The same goes for the next 3 sets for mutation site 32. There was actually bands in our negative control, which meant that there was contamination. So we couldn't trust any of the results in this round of PCR. Once we got a fresh set of primers and template DNA, we ran the PCR again, and we got much cleaner bands as well as no bands in the negative control. We then chose 2 by visual inspection, and we used these to transform the E. coli. This is our sequencing data that we got back from the E. coli plasmid. Site 32's mutation was successful, with the previous sequence GCG being transformed into GTG, as well as the amino acid being changed from alanine to valine. The rest of the plasmid kept its integrity, which meant our mutagenic PCR was successful for site 32. Site 351 was also successful, with the sequence GTA changing to ATA, and the amino acid also changing from valine to isoleucine. So with these newly transformed plasmids, we want to transform yeast with them and apply the high-energy substrate vargulin. We then want to test for how these mutations affected the light production, and we want to repeat this experiment over the remaining 9 amino acid substitutions. So this research, bioluminescence research and Ostracod research in specific, has much greater implications. It can be used for bioimaging and the detection of cancer tissue. The same bioluminescent enzyme used for this imaging is C-luciferase, which is also produced by ostracods. By finding the most effective method of producing the C-luciferase, we can advance this field of medical imaging. I would like to thank my mentor, Nico, Dr. Lena Kim, my TA, Michael, and Aaron, my lab partner. Thank you, and I will take questions.